developed varieties? What kind of approach?
We this time we are importing seeds from these Arabian countries, Iran and Afghanistan, and we are trying to establish that practices because
Mm-hmm.
he is basically grown in the cold desert region.
Mm-hmm.
Uh, it doesn't like rainy areas, so
Mm-hmm.
grows mostly in cold desert regions of Iran and Afghanistan, not everywhere. So we are also having a similar cold desert region in Ladakh and all those regions, near Jammu and Kashmir and Ladakh, and some higher altitude regions of Himachal like
Mm-hmm.
Spiti. So we are propagating there.
Mm-hmm.
And
That's good.
besides that, we are propagating it through micro propagation also. We are establishing that technique also. So it's really a good
Mm-hmm.
that if we get success on this. This is our second year is getting success. First year we have sown some of the plants there in uh,
Okay.
Ladakh and Lahore Spiti regions. Now in second year they again
Okay.
regrow because this is a 5 years crop. After 5 years in the root, we
Oh,
will some this accumulation of this
accumulation.
heme there
Uh-huh.
in the roots. So after 5 years,
Mm-hmm.
so it is a
Mm-hmm.
journey. So for 5 years, taking a crop further is means really a
So then,
you never know.
some genetic engineering there also to
Later
manipulate the
on we
secondary
will try
metabolite
first.
production somewhere.
Later on we will plan that. This time to we are just have to
Uh-huh.
establish it in India.
Mm-hmm.
First is the target.
Uh, but then this is actually a very unique concept. I really liked when you posted it on Facebook.
But after
Because
coming
it
from
is.
JNU and doing these type of things really in the field, it's really
Uh-huh.
pressurized job. Right? So pressure every 3 months. CM asks what how many plants you have grown. Agriculture is there. Standardization.
Yeah.
It's going
It's
on.
actually when the fields are associated, then you have another kind of pressure on your working. When we are doing only lab work related to plant research, then it's something different. When it is in fields, then you have other kind of responsibilities coming up. You have to check out the plants frequently and so many other things. And I think, and as you were saying, the state government is taking so much of effort, so they will be highly interested to know the progress each and every time.
A few weeks back, PM also says in Man Ki Baat that we are now
Oh,
taking in India.
so we
So
are
again,
getting Atmanirbhar on this now.
now the pressure is coming from secretary level and
Uh-huh.
pressure is there. But we have a good
But
here. We have good agronomist team there who they
yeah.
take care of those field
Hmm. Hmm.
good experience in establishing these things.
That's good. That's good.
They are very
That's really
hardworking
good.
also.
Mm.
Oh, okay. So
Okay.
we should start. Uh, just a minute. Welcome everyone. This is our 35th webinar in this series, and we're very lucky. Today, I just told our speaker that today we combine CSIR also, because in this platform we are able to uh, accumulate all the scientists from different backgrounds and different platform and different laboratories in India and international. So this is our first speaker from such a CSR platform. Uh, we are very happy. We are continuing our journey, and it's now 1 year in this platform. We are growing slowly, but our target is slow and steady. We are now also published articles, and few good articles already in published in the online, and many more are in peer review process, and we are very strict about our peer review process. So I congratulate uh, all the participants to join with us. It's an honor to us having a common platform, Plant Science Promotions. Today we have a speaker from Dr. Joshi. our friend Dr. Neha Sharma and Shoma. We are going to host this webinar in a format. First, we are go to the presentation process. We are at the same time we are collecting the questions from participants from the YouTube. We select few questions and to for the live answering process, and then we are doing our interview session where we discuss little more about the speakers and his perspectives on few important ways and methods and aspects. So I request Dr. Neha Sharma to introduce Bioingene and our speaker.
Thank you, Shiva. Welcome to the Bioingene webinar series. The webinar series is a platform from which plant scientists can present their research to the world and future scientists can gain knowledge, perspective, and inspiration. Today is our 35th webinar of the series. We welcome all the viewers who have joined us from around the world. If you are new to Bioingene, please note that after attending today's talk, you can apply for a certificate of participation. For this, you need to submit the feedback form that you have received in your email. The password will be provided multiple times in the YouTube chat box after the presentation. When you fill out the feedback form, please mention the full name of your institute and full institute address. You can collect your participation certificates after 2 days from our website. Bioingene does not send certificates through email. Please visit our website to register for upcoming webinars. You may submit your articles in our Bioingene Plant Science Journal, that is a peer-reviewed open access international journal. We seek to publish papers in various fields of plant science research and address the present and near future scenarios of crop health, yield, and production in different biotic and abiotic challenges. Aspects like food security, nutritional security, effects of climate change, crop resilience, application of artificial intelligence in agriculture, genome sequencing, genome editing, omics technologies, CRISPR-Cas9, modern breeding techniques in plant sciences, etc., will be highlighted in this journal. Selection of manuscripts for publication will be based on novel research, original data, synthesis, or concepts without plagiarism and violation of any copyright. We welcome submissions from all fields of plant science, including agriculture, and from any source. Please visit our website magazine section to know more. You may also send your published posters to be displayed on our website. Please connect with Bioingene via Facebook, YouTube, Twitter, and LinkedIn. So the topic for today's webinar is Genetic Engineering Approaches to Develop Climate Smart Rice by Dr. Rohit Joshi.
So shall I start?
Institute of Himalayan.
Uh, Neha?
Yeah.
Okay. No, yeah, actually, you stopped somehow. Okay, it's okay.
Dr. Sorry. Dr. Rohit Joshi is a senior scientist at Department of Biotechnology, CSIR-Institute of Himalayan Bioresource Technology, Himachal Pradesh, India. He obtained his Ph.D. from GB Pant University of Agriculture and Technology, Pantnagar. He carried out his postdoctoral research at Indian Agricultural Research Institute, India, Louisiana State University, USA, International Center for Genetic Engineering and Biotechnology, India, and Jawaharlal Nehru University, India. He has 14 years of research and teaching experience in genetic engineering approaches to improve tolerance in plants against individual and combined abiotic stresses, and has published about 50 peer-reviewed publications with total citations of more than 1,500. His current research interest is elucidation of abiotic stress tolerance mechanism in plants. Dr. Rohit Joshi has received the Young Scientist Award by Indian Society for Plant Physiology IARI New Delhi, R.D. Asana Gold Medal Award by Indian Society for Plant Physiology IARI New Delhi, and more. Last year, he was elected as a member of the Plant Tissue Culture Association of India. He is a life member of many renowned scientific societies of India. Dr. Rohit Joshi served as a reviewer for many international journals like BMC Plant Biology, BMC Genomics, Frontiers in Plant Science, Journal of Experimental Botany, Algal Research, PCTOC, Plant Science PLOS One scientific reports. We are highly honored to have him today on our platform. I now request Dr. Joshi to please share his screen and begin his talk.
So, am I audible?
Yes, sir.
And my screen is visible, yeah?
Yeah.
First of all, very good morning to everybody, respective in the evening to other parts of the country. Maybe some of them are from other parts of the country. So, first of all, I would like to thank the entire Bioingene team to give me an opportunity and a platform to present my research that I have done in my previous years. And today I am presenting on my topic, as you all know, that genetic engineering approaches to develop climate smart rice. So, as we all know that
Sir, your screen is not shared yet. So please share your screen once again.
more. No problem. Sorry. Is it visible?
Yes, yes, it's visible.
Mm.
Okay, carry on.
As we all know that climate change severely affects the biomass productivity and yield of crop plants, and it negatively affects the plants survival and yield. So there are several plants show several stress responsive mechanisms to cope up these abiotic stresses. And in underfield conditions, we observe that initially we thought that we work on the previous years, we work on individual stresses. Now, slowly the people came to know that in underfield conditions, none of the abiotic stresses are coming alone. So there are multiple stress conditions coming together. And when we deal plants under field conditions, so now the scenario is changing to deal with the simultaneous and stresses that are coming together. So I will now. What is our current understanding of these multiple abiotic stresses? So, just to taking an example here about the drought stress, we can see that there are there are several mechanisms or several pathways, and their genes regulating those pathways. They interact and they overlap each other to like as orchestrating about the key molecular switches in past years and in current scenario, we are unraveling these genetic architecture of plants to delineate these pathways overlapping with each other. To find out what is the basic mechanism behind how the plants deal towards these abiotic stresses. So now the omics approaches are coming. So these omics approaches, using different omics approaches, we are now targeting to different molecular networks, and then also we are identifying how these network really means interact with each other and play to in an orchestrated manner to provide tolerance in plants. So for this various approaches we can use to develop stress tolerance plants. But as we all know that in agriculture in the 21st century faces multiple challenges. Now there's abiotic stresses. There are several abiotic and biotic stresses. They are imposing major constraints in crop yield. So now there is a transdisciplinary approach is required to solve these complex multigenic traits. Like we have genetic engineering on one hand, and then bioinformatics on other hand, and then we use mathematical modeling to identify these plants, and to further identify the physiology and biochemistry. We have to understand first to do some molecular work or to target some genes, and then we come to the system biology, and whole life science is there that plays a major role. So every branch is important when we are dealing with the transgenic plants to develop those transgenic plants which are really climate smart. So we have to take all the branches together. So now the thing is coming that this transdisciplinary approach is very necessary to develop these transgenic plants. So one approach to develop these means draw abiotic stress tolerant plant or high yielding plants, that we can say the climate smart plants, is a conventional technique which is a long process of selection and backcross breeding. But there is another approach is the molecular stacking, or what we say that the molecular biotechnology, through biotechnology approaches, where we overexpress or knock down some of the genes to delineate their functions, and then we can develop the tolerant plants that are some or those genes which are the key regulators pathways, and we can develop the tolerant genotypes out of that. So taking into account that, we know first this salinity stress is a major environmental challenge that affects the cellular homeostasis and ionic toxicity, and thus limits the crop productivity. So in our major experiments, we use this Pokali rice. Pokali rice is basically a flood and saline tolerant rice variety, and it is cultivated in India in the waterlogged coastal regions, and it attains a height of almost 1 meter, and that's what it is. Why it is flooding tolerant also, and it is because it is grown in coastal areas, so it is like salt tolerant variety. And now this brand Pokali has also received a GI tag, and it is considered as a highly valuable gene donor in various international rice improvement programs. So first of all, if we came to when we are dealing with the salinity stress tolerance, or we can say the multiple abiotic stress tolerance plants, we came to know about cyclophilin gene, which is ubiquitously present in all the subcellular compartments of the of plants, and we isolated this OS as CYP2 from Apokali, so named it as OS CYP2P, and it is isolated from salinity tolerant landrace Pokali, and we ectopically express this OS2EP in tobacco plants, and we found that these transgenic plants become tolerant to multiple abiotic stresses like salinity as well as drought. So if we will see that under different salinity levels, we found when we analyze these transgenic plants, we found that these transgenic plants show less bleaching of chlorophyll than their wild type counterparts and in under salinity conditions. So these are flourishing very well under these salinity stress conditions. So besides this, we also found that the transgenic lines showed reduced lipid peroxidase content and reduced electrolyte content, and the reduced superoxide content under the stress conditions like in salinity or heat or drought, and the osmotic stress, if we will observe, And they also showed, besides this, they showed higher chlorophyll content. You know. So from this we came to a conclusion that this cyclophilin gene operates via ROS scavenging and ion homeostasis and enhance the abiotic stress tolerance in plants. So this is one gene that we have used in our study to develop salt tolerant or dot tolerant um, right transgenic plants. So further we move to another gene that is a multifunctional gene because this is a phosphoglycerate kinase enzyme that plays a major role for ATP production during glycolysis and 1,3-phosphoglycerate in the Calvin cycle in plants. So this because this plays a role in glycolysis and Calvin cycle and other pathways also it is involved. So it is called as a moonlightning protein. And so we focus on this, and we found while analyzing in the contrasting rice cultivars IR64 and Pokali, we found its higher expression in under salinity stress conditions. So this OSPGK2 transcript and protein showed that it is strongly induced by salinity stress, and so we isolated this enzyme from this gene from Pokali and also from Pokali, and then we cloned it and transformed it in tobacco plants. So we found that ectopic expression of transgenic tobacco improved salinity stress tolerance in this. And here we see that another time you see that while overexpressing this OSPGK2 EP, the transgenic line showed higher chlorophyll retention than their wild type counterparts, and also they showed the high proline content also. And in addition to this, the transgenic lines showed taller phenotype, and also they are having more pods. So obviously they are having higher yield besides the abiotic stress tolerance. And under drought stress, we found that their stomata are less closed, so they are still having a tolerance against drought stress besides salinity stress tolerance. So this is one approach. Another approach to develop the transgenic lines that really survives under salinity and drought. So further coming to another gene that is a metallothionein gene. So earlier these metallothionein genes these were recognized for providing tolerance against heavy metal response, and they are basically named like this metallothionein gene because of their heavy metal responsive function. So these are low molecular weight cysteine rich metal binding proteins. We isolated this gene also from Pokali, and we transformed it in tobacco. And before that, when we do the transcript analysis, we found that whole genome analysis, we found that these metallothionein genes show higher expression under various abiotic stresses besides heavy metal stress in our study. So we cloned it, and we found a positive correlation between ectopic expression of OSMT1EP isolated from Pokali and the abiotic stress tolerance. So we found that different transgenic lines overexpressing OSMT1EP showed higher germination rate even under very high levels of salinity, as well as under metal stress like copper and zinc, and also the polyethylene glycol induced dehydration stress. So when we observed their chlorophyll content under different stress conditions, we found that the transgenic plant showed less chlorosis under multiple stress conditions like heavy metal, cold, and polyethylene glycol mediated dehydration stress, also the salinity stress, and as well as in heat stress. So in comparison to their respective wild types, where you see the complete bleaching was observed. So besides this, we also found that they are having less accumulation of reactive oxygen species under all these various stresses in comparison to their respective wild type plants. So this is a very nice study where we found that a transgenic plants having tolerance towards multiple abiotic stresses simultaneously, and so we found that this also functions through this provide tolerance to this basically through a scavenging of reactive oxygen species. So coming to another approach where we see that in our previous studies we are dealing with the Pokali rice. So further we found that there are some halophytes which are really salt tolerant, and these halophytes, they grow very well and flourish very well, and they produce viable seeds, and they complete their life cycle in high saline conditions. So we are curious to know about whether these halophytes, can we isolate some genes from these halophytes to increase the tolerance levels in rice plants through molecular biology or genetic engineering approaches. So we have one halophyte that we came to know is Spartina alterniflora, which grows in the coastal areas of USA, and it grows in very high levels of salinity stress. So we isolated one gene, that is ADP ribosylation factor, which we call ARF. These are regulators of intracellular vesicle trafficking, and during various abiotic stresses, they produce various secondary metabolites like phosphatidic acid and PIP2. And so we isolated this ARF gene, ARF1 gene from this Spartina alterniflora, and overexpress it in rice cultivar IR64. And then we found, then we have both in IR64. So during our physiological analysis, we found that similar to our previous approaches, it also showed less chlorophyll bleaching, or we can say the higher chlorophyll maintenance than their respective wild type under high level of salinity. And also, if we similar simultaneously we transformed in Arabidopsis lines also, and we found that these Arabidopsis lines also showed higher salinity tolerance, and they are having more profuse rooting as well in under high saline conditions, that is 150 millimolar NaCl. And similarly, these rice transgenics, when we grow, these also show higher growth as well as higher root growth and shoot growth both, and under a high level of salinity. So besides this, when we grow these transgenics in under drought conditions in Arabidopsis and rice, we found also there also that wild type plants they died after a few days, but these transgenic lines show very profuse branching and shooting. They are even in a very severe level of drought stress. The similar thing we observed in rice plants also, where under severe drought conditions we see that these wild type lines almost they died, and but these transgenic lines, they survive very well under drought conditions. So we analyzed their physiological analysis, and we found that they really maintained high chlorophyll content um, and very less electrolyte leakage during these salinity as well as drought stress conditions. So another approach is that we isolated another gene that we termed as actin depolymerization factor, which maintain the cellular network dynamics. And we found in this experiment what we have done, that we isolated this ADF2 both from the halophyte spartina alterniflora as well as from rice vocali. And then we transform both of these genes that we isolated from spartina and from vocali to IR64. And we observed a very surprising differential response in the transgenic developed from the gene of from Spartina alterniflora as in comparison to the gene that was isolated from rice. So both of these genes were transformed in IR64 rice cultivar, and so we found that these that were developed from overexpressing SAADF2 showed more profuse growth even than the transgenic developed from the rice OSADF2. However, both of these transgenic lines showed better response than their respective wild type in shooting as well as in rooting. But in comparison to both of these genes isolated from spartina and rice pokali, they show differential response in between them. So we try to delineate how why this is showing different responses. So we analyzed their amino acid structure, and we found there is a difference in few amino acid residues, especially serine residue that blocks its interaction with CDPK6, and that really decreases its decreases the tolerance of transgenic plants over expressing OSADF2 in comparison to SAADF2. So so this is one thing. And another thing that these transgenic lines they are as previously known that they are having higher relative water content, and they are having a higher membrane stability index besides having higher proline content, and the plant height, they are having more higher plant height, and as well as the fresh biomass they have attained. In addition to this, when we have done the anatomical analysis, we found that ADF2 that transgenic plants that are developed from spartina ADF2, they showed intact grana during the prolonged drought stress. However, these the transgenic plants overexpressing OSADF2 from rice, they also showed drought tolerance, but not that much prolonged as compared to Spartina, the transit plant that are developed from Spartina alterniflora gene. So this is really a new study where we say that there is something difference between the rice wild type rice and when we take the real halophytes that are really surviving under high salt concentrations. So again, when we do this chlorophyll leaf disc assay, then we found again then these during high salinity stress conditions, they show very less chlorophyll bleaching, and their survival rate is very well, and they produce the seedling growth is very good under high salt concentration in comparison to their wild type counterparts, and they are having higher relative water content in comparison to their wild types, and they are having higher stomatal conductance levels. So from this we came to conclude that SA spite alterniflora ADF2 overexpressing conferred salt tolerance in rice plants in a much better way. So then again the theory came that besides abiotic stress tolerance, the plants must have higher biomass and yield also. So sometimes we see that many of the reports are there that when we developed the transgenic plants, they are tolerant to abiotic stresses, to several abiotic stresses, but yeah, simultaneously they are having very less yield. So we had to now the approach came in our mind is that why not we develop those plants, transgenic plants that are abiotic stress tolerance as well as they are having higher plant biomass and yield. So we focus on this area and we found that there is a complex network again, like previously we have found in the abiotic stress, there is a complex phenomena, but similarly to that also in during biomass and when we compare the complex network of biomass regulatory gene and abiotic stress regulatory genes, so they are also interacting in some or other ways, and there is a complex signaling network for biomass production, abiotic stress tolerance we observed, and we tried to unveil the crosstalk between these transcriptional circuitries for biomass production and abiotic stress response. So another idea that came to our mind for having these interacting pathways, one pathway we observed that there are phytohormone responsive pathways. So these phytohormones are somewhere responsible for regulating plant growth and development, and also they promote the survival and acclimation towards stress. So we go deep in this network, and here the figure which I am showing there, we found that there are several these hormones that they are affecting various plant growth and promotional activities besides providing stress tolerance in plants. So keeping this factor in mind, we as we know that the whole world is now focused on the crop improvement programs to enhance grain yield. So we now this time we go for another approach, that is gene silencing. So post transcriptional gene silencing, as we observed, is a constitutive complex gene regulatory defense mechanism functional mostly in all eukaryotes. And when we found these small RNAs under dealing with the biomass and the salinity as well, then we found there are some common factors that in between both of these traits. So we focus on this, that why not to focus on these to improve the abiotic stress tolerance as well as yield. So we came across one enzyme, that is cytokinin oxidase, which catalyzes the irreversible degradation of cytokinin, and then this catalyzes the irreversible degradation of cytokinins, and thus we to increase the cytokinin accumulation in plants, we go for another approach, that is gene silencing. So we silenced this CKX2 gene in IR64, and we found that when we developed these transgenic lines, some of these lines show very less activity of CKX2, even below 70%. So these line we take for further analysis, and we found that these This knocking down of this inflorescence meristem specific rice OSCKX2. This showed negative correlation between its expression and plant productivity under salinity stress. So we have done two experiments simultaneously, where one experiment in which we get a complete big block box where we put we grow these wild types and these transgenic knockdown transgenic lines of CKX2 together. So we found that the growth is very high in these transgenic lines in comparison to their respective wild types. And when we compare the yield of these transgenic lines with their respective wild type, we found that these transgenic lines showed profuse growth under salinity stress in comparison to their respective wild type plants. So further, if when we check the PANICLE architecture of these transgenic lines in comparison to their wild type, we found that we found a higher PANICLE branching in the knockdown transgenic lines in comparison to wild type IR64. And when we see the physiological analysis, we compare that they are having much higher chlorophyll retention. Also, they are having higher number of panicles, and the grain weight is somewhat on the higher side in the transgenic lines. And they are having under salinity stress. When we observe that, then also we found that these transgenic lines showed a higher number of filled grains per panicle, because we know that during salinity stress, even though the plants produce panicles, but they are mostly chaffy grains. So to check those, we done this experiment, and we found that higher number of filled grains per panicle, and also leading to this high harvest index rate in the transgenic lines. So this is a real good approach to develop transgenic lines, and besides having higher abiotic stress tolerance and higher yield. So so to check how this panicle branching that occurs in these transgenic lines, we check the cytokine oxidase levels and its activity in 8 different stages of panicle development in both the wild type as well as transgenic lines. So we see that they are the CKX2 is differentially regulated in the panicle developmental stage specific manner, and it is having very highest activity during the 13 days after panicle initiation, from where the panicle branching starts. That's why it has having higher panicle branches in the transgenic lines what we get. And from this we can conclude that OXCKX2 via controlling cytokinin levels regulates floral primordial activity, modulating rice grain yield. So coming to another thing that came later in our mind, that we found that though we all know that the contribution of biotech crops to food security, sustainability, and climate change solutions. So these biotech crops are having a good impact, and in various developing and developed countries in their food security and sustainability and climate change solutions. And we are having we found several advantages of these biotech crop genetically engineered developed crops in comparison to their respective wild type crops. But in certain countries like India, there is still these antibiotic marker based selection methods have several biosafety concerns. So they are not getting approval for cultivation of these biotech crops and for public welfare. So Another hand, there is a less public acceptance in between the common man also, because of lack of knowledge about these transgenic lines. So for better public acceptance, and in order to promote commercial deployment, we focus on the marker-free transgenic lines. So to develop these transgenic lines, we have chosen free halos as a metabolite. So trehalose as a basically a non-reducing disaccharide and signaling metabolite also. So its biosynthetic process is a two-step enzymatic process utilizing two enzymes, that is trehalose phosphosynthase and trehalose phosphate phosphatase. Both of these enzymes were previously been reported to provide abiotic stress tolerance in various crop plants. So our idea is that too, why not we fuse both of these enzymes together to make a single construct to develop the transgenic lines. So we make a fusion construct of both this trehalose 6 phosphate synthase and trehalose phosphate phosphatase gene. So we named it as a TPSP, and this we overexpress in again in rice IR64 cultivar under the control of an ABA inducible promoter. So we found the higher accumulation of trehalose metabolite in the transgenic lines in comparison to their respective wild type plants. So after overexpressing this TPSP gene in IR64 rice transgenic lines, we found that during seed germination stages, we checked what is the effect on the seed germination rate. So we found that these transgenic lines can tolerate both the high pH level, that is up to 9.9, and high EC electrolyte conductivity level, that is around 10 decisimals per meter square, and also the severe drought, that is around 30 to 35% soil moisture content. So so we grow these transgenic lines as transgenic seeds in under salinity, sodicity, and drought stress. And we found that these transgenic lines really perform much better than their respective wild type plants, and they flourish very well. So when we check they are having a high germination percentage under the sodicity, the salinity, and as well as in drought. And when we compare the shoot length and root length of these transgenic lines, then we found that these transgenic lines show higher shoot length as well as higher profuse rooting. You can easily see in this figure that they are having profuse rooting also under all these salinity, sodicity, and drought stress conditions. So then we check these plants again in the vegetative stage and the reproductive stage. Then we found that these transgenic line will with high trehalose content maintain higher yield, as well as they can survive under various high level of abiotic stress conditions. We check these plants in under salinity stress levels and sodicity levels and drought stress levels. So we found that that the transgenic lines overexpressing TPSP, they showed profuse growth as well as higher yield under these all of these abiotic stress conditions. So we found that they are having higher trehalose accumulation also under these abiotic stresses with a higher chlorophyll content they have maintained during these abiotic stresses. Even though their salt accumulation level is very well, and they maintain their respective relative water content levels very high in comparison to their respective wild type plants. So as we all know, this sodicity, the term which I have used, that this is a major problem these days in India and other major countries. So this is very good approach to develop a transgenic lines. We have developed for the first time against these sodicity stress conditions. After this stress for 25 days and for 60 days stress, we have found that these plants are showed very profuse growth. Even in sodicity stress condition, when we go for their recovery after that, then also we found that these transgenic plants recover very well after when there is a control conditions being applied. So we can see that from this that we can see that these not only these plants can tolerate a longer duration of these abiotic stresses, and besides that when the normal conditions will arrive, they can revive very well, or they can revive much faster way in comparison to their respective wild type plants. So from here we have developed several approaches. Now I am going to other CRISPR-Cas and other approaches to deal with these plants. Finally, I would like to thank Professor Dr. Sanjay Kumar, our director from the institute, for providing all the necessary facilities for research in the institute. And I would like to thank Professor Sopuri for their eminent guidance, and also to my mentors, Professor Ashwani Parikh from JNU, who is now the director in Nabi, Mohali, and Dr. Resne Lata Singla Parikh. Besides that, I would like to thank my mentors from IRI, that is Dr. Vishwanathan Chinnuswamy and Dr. Niranjan Mesak from Louisiana State University. And finally, I would like to thank so much to Dr. Subrato Mazumdar, who gave me an opportunity to present in this, who sent me invitation and allowed me to present in this platform. So thank you all. Sir, I cannot hear your voice.
Hear me, sir?
Yeah. Now it's okay.
Thanks, sir, for your nice presentation. It's lots of work, and most interesting part of you choose a variety and isolated gene and analysis all the related pathways of that. It's encouraging for those who are working on transgenic and non-transgenic. Both of sector is very interesting, because genes are available if you wanted to know the function, and you can create a better product. And we got so many appreciations about your talk, and we selected few of questions for you. Those are related to your talk, and obviously, so when you change something, there are a lot of anxiousness created. How happened? How you use? So let's focus 5 or 6 questions. I pasted it in the chat box, so we can start from that. If we collect few more, then we ask you once again after your interview session.
The first question is Pokali also heavy metal tolerant, and what makes you choose metallothionein from Pokali and not from arsenic tolerant species like Triguna rice? Very nice question. Very nice question. It's really a good question. Why? Because why we have choose metallothionein? This is very interesting story. That while this metallothionein genes, as I have told earlier also, that these were reported basically for heavy metal stress tolerance pathways, because these are basically heavy metal regulators genes. So, but surprisingly, when we are doing the preparing a subtractive cDNA library in our laboratory, so not me, but my previous workers who have worked on this subtractive cDNA library, they found out surprisingly that some of these metallothionein genes, they are having higher expression in tolerant rice cultivars like Pokali. So from there, we got an idea to why not we check these metallothionein gene in our study also, whether they are providing stress tolerance really or not. So first of all, we have repeated this subtractive cDNA library, and then we found the similar results, and then we produced these transgenic lines, and while producing these transgenic lines, we found that these transient lines are really producing means they are having higher abiotic stress tolerance, both heavy metal for drought, for heat, and for salinity. So this is a very nice approach what we get out from that subtractive cDNA library. So another question is, why not to take from some arsenic tolerant species? So at that time when we are dealing with metallothionein metallothionein genes to overexpress in our rice cultivars, we are not dealing with some heavy metal tolerant developing some heavy metal tolerant rice. We are basically dealing and in developing a abiotic stress tolerant, means basically to salinity, much focused on salinity and drought stress tolerance. So we are developing a cDNA library, or we are checking at that time the genes those are responsible for somewhere for salinity and drought stress responses. So by discovering or characterizing those genes, we came across these the metallothionein gene that is also in Pokali. So we choose it from Pokali, and they really perform well. But the question is good for if we want to develop some specific stress tolerance arsenic, then we can choose the gene from like these arsenic tolerant species like Trigona rice. This is the study what we have done can open the arena for other researchers also. They can use those approaches, and they can develop several other abiotic stress tolerance rice varieties. So next question is that you have taken genes from Okali rice cultivar and overexpressed in rice. Okay. Will you get different phenotype if you take genes from salt susceptible rice genotype and overexpress using strong promoter? No, this is a generally general question when why we choose these promoters, because promoters generally usually in our previous studies we are using these constitutive promoters, and these constitutive promoters at that time and later on the studies, the people found out that these constitutive promoters, why you use constitutive promoters when you are dealing with some salt tolerance, or you are dealing in some means stage specific manner, or some this yield or some fruit specific. These days you are things you are dealing. So that also, this thing also came in our mind later on when we are doing experiments. So this is the story what I am telling was from step by step how we changed our approaches according to the theories came, and how what are the requirements further for that. So later on we changed our promoters also in our later studies, and we studied several promoters also. We identified several promoters also that I have not present here. This is the good thing that even if we are using this constitutive promoters in our study, we do not found any change in phenotype, except but we can say that change in phenotype in respect to if you are asking about the profuse branching or more growth, then we found that yes, there is a good yield and growth we found. Another question from Joy is, why is tobacco chosen for ectopic expression of the trait? Tobacco, we have tobacco is basically we used as a model plant. So in our initial studies, we are taking There is a tobacco plants for overexpressing these checking the functional characterization of these genes, which we are taking in our study. But further, as we grow through our research, we several of these people question about these things, that why do you use under means you ectopically express those genes. Why not you express those genes in rice to develop some tolerant rice cultivars that people can use? What is the use of developing tobacco? So we later on we changed, as I have shown in my study also. Later on we shifted to rice. Now from we take the genes from Pokali rice also, and then we transform it in IR64, that is a sensitive cultivar. Even though we have taken genes from the Spartina alterniflora, that is a halophyte, we also transform that in Arabidopsis also for our quick mechanistic survey, and for in IR64 also to check their real expression or real functional characterization in a sensitive cultivar. So this is the thing what we have done. So next experiment question, sorry, is sir, which gene or genes in biomass production are most responsible for handling abiotic stress responses? So there are several genes, because as I have shown in my some of my slides, that this is not the gene what are really responsible for handling abiotic stress responsible or not. There is a crosstalk. So sometimes we while dealing with the genes, we come to some key transcriptional regulators or some transcription factors. They are key genes, or some genes that they also act as a the key for to regulate several other genes downstream. So those are the gene when we use we can simultaneously regulate. When we regulate those genes, we can simultaneously regulate several other genes also, because as I have shown in my slides also, that when we found that the different abiotic stresses interaction, then we found there are several genes. There is a complex regulatory network of several genes and between these various abiotic stresses. And then when we compare to these genes, some of these complex regulatory pathways of the genes regulating these abiotic stresses and also the higher biomass. So then also we found there is a complex regulatory network. Another complex regulatory network is there, that is dealing between biomass also and in abiotic stress tolerance also. So some of the genes are common there, some of the genes are common here when we are dealing with multiple abiotic stresses. So mostly we take some common genes that are when we are speaking about multiple abiotic stress tolerance. So we take some common genes that are that can provide tolerance to multiple abiotic stresses, because in field conditions none of the stress come alone. So there are many of the stresses come simultaneously. And similarly, when we are dealing with biomass, so we have to deal with the biomass also. We cannot develop those transient plants that are tolerant to abiotic stress, but do not have yield, because yield is the major factor, because the population is rising. So we check some common genes that are more that are the key regulators basically. Another question is, can you share some screening protocol for salinity and drought? Yes, you can mail me. We have developed some protocols for this screening protocols, and also in there are several books also they are published that deals with with different protocols they have developed for screening the salinity and drought stress response. Basically, one or two book chapters we have shared in CABI journals. They usually have a very beautiful screening protocol for analyzing this salinity and drought stress response in various crop species. In different crop species, they have standardized. Like this ED, if you will take in rice, ED has their own protocols. They have fixed protocols. You can get it from their knowledge database. You are going to ED website and go to their knowledge database. You can get. a very good protocols there for screening of salinity and drought stress response. Um, Another is, what are the major constraints in abiotic stress tolerance rise? So, as I have told earlier, also the major constraint initially in our initial studies, we are dealing with the salinity stress response. Then we go for drought stress response. Then the question arises: then what about field conditions? When you will go for field, the multiple abiotic stresses come together. What you are doing for that? If you develop a salinity tolerance cultivar, whether it will survive in the drought stress condition. uh, this question arises, and then we shifted our studies to multiple abiotic stress tolerance. Then another questions available arises, that whether you develop multiple stress tolerance cultivars, whether these are having higher yield also, because people need food. So again we shifted, take that opportunity, and we shifted to another approaches, and then we developed a biotic stress tolerance rice cultivars with high yield. Then also again the question arises, who will eat that these transgenic rice line, that transgenics are not, people are not taking, because they are having these selective markers, and those people are not interested in those markers. So we will develop another marker free transgenic lines also. So now again the CRISPR Cas is coming. So now the whole genome you can edit, rather than ectopically expressing genes from other cultivars. You can check directly. So now the approaches are slowly changing from when I started my these genetic engineering approaches past 15 years back. Then the approach is different. Now after now if you will go to the current strategy, now the people have changed their strategies also. The problems are different. People have a more good understanding. So this is the main thing, that when we are doing research, so slowly we will develop our understanding. Problems will we do one thing, and then some problems will arise, and we'll cope up all those problems, then another problems will arise. So the scientist work is to means to answer those questions that are coming through farmers or different other peoples, that during their study. So this is what thing. Um, so another approach question is, has there been a variety developed that incorporated the different stress tolerance traits? Yes, we have developed transgenic lines, but unfortunately in India the transgenic approach is transgenics are not allowed in India, and other countries they are. So probably in future, so after some time we will be able to develop some. So we have another approaches also now. My mentors, Professor Parikh has doing a good approach in that they are doing some developing some mutants, and they are analyzing, and they found that the mutants that they can directly release in the for the welfare of public, if they found. And some breeders are also doing. So there are different approaches. Basically, the basic thing is to increase the productivity of to feed the increasing population. So any approach we can use. Rather, we can use the transgenic approach. If you do not believe on that, then go for the breeding. Traditional breeding approaches people are also developing now these days. The fast breeding approaches are coming. So from there, people are also developing very good research in this respect going on. One of our good Professor Rajiv Vashne, they are doing very good in legume research. No? We all know. So every different scientists are going through different approaches. The basic goal is to increase the productivity for farmers. Any approach we can use. Just we have to believe in something. So another question is, there are so many good products of rice in the pipeline developed by transgenic approach, but they are not yet commercialized. What may be the reason? So there are some regulatory features. Also, no? Government has developed some guidelines. So government has to develop some guidelines because government has a priority for human safety, safety of the common people. We cannot do any haphazard research and can release in the market directly. So government has developed some guidelines. There are also big scientists and learned people there who prepare these guidelines. So So we have to believe on them also, because if they are preparing those guidelines, then those guidelines mean something. And we cannot bypass all the guidelines, and we cannot do whatever we want to do in our research field. So there are certain guidelines we have to overcome those the guidelines. They are actually, these guidelines are the pathways also. So how we have to change our strategies, what is really to be for public, what is not. So we have to change our strategies according to, we can develop something for public. It's not the thing that research they haven't blocked. People are doing research also. Research government has not blocked. But besides having some developing the high productivity or yield or enhancing yield, the another part of research is to understand the mechanism also. So there are several research previously have done, like in photosynthetic pathways, in respiratory pathways. People have done so much research. Then the whole pathway we know today. So people are working for different mechanisms and pathways also, so that people can get the good understanding on the mechanism also, so they can use in their research in other ways also. So like we developed some novel pathways, and then if the transgenic approach is not there, then breeders can use that approach and to select those genes or the QTLs to transform through traditional breeding, and then people can use that. So uh, uh, one another question is, thanks for the presentation. Can this gene also be used for transformation of maize and underutilized legume against abiotic stress tolerance? Yes, yes. Yes, why not? As you know, the science has no boundaries. So if you have, if I am talking something that this gene is favorable in rice, in providing abiotic stress tolerance, that gene can be then can be applied for other crops also. We are also using something that some studies initially came from Arabidopsis plants, then people will try them in their crop species. So this is what the research is going on. But the major thing is that first you have to understand the psychology of plant. You have to understand the basic physiology or the biochemistry of the plant. Whether before inserting any gene, whether that gene will really work on that crop or not. And just overexpressing any gene doesn't mean anything. You know? Means because every plant has a different pathway, and some plants produce secondary metabolites, some plants produce good flowers, some plants are having higher yield, some we eat, some we do not eat. So there are we are different plants, we are dealing with different strategies and different things. We know every plant, we are not dealing for this abiotic stress or high yield, because everywhere there is no problem for this abiotic stress and higher yield. Some plants are for more flowering behaviors, and some plant we need more flowering behavior, some plant we need higher biomass. So we have to focus on those things, what exactly our target is. So next question is, the useful genes were sourced from rice and put into rice. So what is the transgene in this scenario? Yeah, this is a very good approach. This is rather we see that the approach is the same, ectopic expression, because it is shifting from one species to another species. But is the main thing is that when we are because of public understanding, that's what I am saying earlier also. So we have to change our strategies from for the public acceptance, and to provide that thing that are useful for society and not for just for research and fun. So the question arises there, that when we are taking genes from other, suppose we are taking genes from other organisms and then transferring in plants, and people say that, oh, that gene is from this animal, or that one we will not eat because it is not like that. And so we change our strategies. Earlier we are transferring some genes from E. coli also, then we transformed genes from other halophytes also, then other things also. Then people suggest, then the question means arises several times that Why not you take from a rice gene and transform it in rice? That will be more safe because it is already available in one rice cultivar. So taking that genes from one cultivar to and putting them in the same cultivar, that is that you really can to increase the sensitivity of increase the tolerance level of those sensitive cultivars also. So this is like the crop improvement type of program that we first characterize that gene in the respective tolerant cultivar, and then we transfer that gene into its respective sensitive cultivar to improve that crop variety. That approach we usually do in the breeding approaches also, but this is a different way to develop that crop improvement. So another question is, sir, are you interested in collaborative research for development of transgenic cotton from Pakistan? Basically, this is a good question. Yeah, and the science is always a collaborative work. Several people are doing collaborative research these days, and yes, the collaborative researches are always fruitful. So basically, I am not this time doing work in cotton, however, but yeah, you can approach some. There are several cotton breeders or cotton biotechnologists that are doing research in India also. But the good thing in this respect is that the cotton transgenics are allowed in India. So you can do very well with the Indian collaborators when you are dealing with the cotton plants. So it is really you can get some output there also rather than doing other crops.
Thank you, sir. I'm not pasting any more questions. There are a lot of. I will email you the rest of the questions.
Okay.
So we are now going for the interview session. I request Shoma to do an interview, and lots of young researchers are waiting for that. Thanks.
Hello, everyone. Ashoko, could you just switch down the volume of your laptop? Okay, great. Thank you. Hello, everyone. You are watching Dr. Joshi's very enlightening and informative talk on genetic engineering approaches to develop climate-smart rice. We will now move on to the interview section of today's event. Hello, Dr. Joshi. We are very lucky to have you with us today. We will take this opportunity to know more about you. Sir, we would love to know how you built your career. Could you please share your journey as a plant scientist with our viewers?
So my journey towards this science started from my childhood days. As my father is also a scientist, Dr. P.C. Joshi is a scientist, and he's a deputy director at that time in Ayush Ministry of Ayush, and he's basically a chemist, a chemistry background. So he does a good research at that time, and he has identified several new compounds. While doing his research and while doing his PhD and the post PhD research during his job, he identified several compounds and that are very useful these days also. So that are well recognized compounds. So from there, in my childhood, I got an idea that yeah, the science do something in society because I see that thing by my own. So later on, this my studies are continuing like everything and else, and then slowly I came to this after during my PhD. Also, my guide is Dr. Late Professor Alok Shukla is a good scientist. He really does a good work, and he always have come up with new ideas and share with him. And completing my PhD, very luckily I got an opportunity to work in with Professor Ashwini Parikh and JNU directly after my PhD. That is a real turnover in my research thoughts and plans. So he's a very good scientist, and he molded my mind towards developing my research career. And there I have developed most of these farming plants I have developed there. And then after that, I went to IRI, and then I worked with when I worked with Professor Vishwanathan Chinnuswamy. There also he is having very excellent research background, and I was amused to see his research publications and research at that time. So so that all things slowly developed in my mind that I should focus on research and develop my research career. So that I went to USA, and then there is a different working environment is there, but there also I learned a good professional research experience. And there also there I worked in that Spartina, alterniflora, halophytic genes, what I was so then I returned back to India, and then joined with Dr. Snehata Singla Parikh, who is also a eminent scientist. And also there I also get in contact with Professor Supuri, who is a well-renowned scientist in India and top scientist of India. And he really opened my mind in different aspects. And when we worked together, we came across with several good research research. So slowly means the time I learned a lot from them also. And then I finally took up my career as a I think that I should go for research career. And then I started writing my own grants, like DS Kothari and CRV young scientists I get. And then finally I joined our current director gave me an opportunity to join this institute and work here for this high altitude Himalayan bioresource. So this is a good opportunities. And while in journey I learned a lot from all those learned people which I have joined. But I feel lucky that because I'm lucky enough to have joined with those people in my early career development yesterday. So they shaped my career in a good way, and develop my understanding in a much deeper way.
All right. What motivates you to work for the betterment of agriculture?
So basically, I am from agriculture background, and I have done my PhD from Pant University of Agriculture and Technology. So it is a very premier agriculture university, and it has developed several varieties. Even I have my co-guide and my guides. They have developed several varieties that are released in the market, and there is well known on university to develop several varieties that are grown in that Ganges, Indo-Ganges belt. Several rice, wheat, maize, and they have developed several cultivars for that region. Not from for UP plain region, also for high altitude Himalayan region for Uttarakhand as well. So from there and after while doing my postdoctoral work in IRI also, that IRI is also a premier institute who has developed several varieties. And there also I get several ideas that you have to do some research for public prospect to develop some cultivars that can really be used by public. So and there is an annual function where they are released these varieties. And there is also the Kisan Mela organized in IRI and Pantnagar also. So that are really mind-opening things where the farmers really ask about their questions, real questions. They ask what you are doing for ourselves, what you are doing for farmers. You are really ready, really flourish well in field or not. They directly ask the questions. So while answering those questions, the scientists also have can open their mind, because they are real workers who are working on fields. So they are just the booster for our research program, that they ask the question, and then we try to find out their answers.
Do you think we should consider abiotic stress more seriously to ensure global food security during the times of climate change and global warming?
Yes, we can say this is a good approach, because the prediction is like that climate is changing in a high speed. And for in coming years, 50 or 100 years later on, we will see a different climate than what we are seeing here. This may be a general prediction also, but this can be true. We do not know what is hidden in the future. In the future, so we have to. The only thing is that we have to prepare ourselves rather than sitting that when the climate will change, then we will see that what we can do for that. So the scientists are just preparing developing some cultivars and preparing themselves that when this suddenly the one day the climate change will occur, and someday we will found that the abiotic stress problem really comes to our field, neighborhood or to our fields, then what will we do at that time? We are not going to search for those tolerant cultivars or developing those cultivars overnight. So
Yes.
we are having different approaches there. So as we see in our research also, that while developing these transgenic lines or these climate smart crops, we will see that different new questions are arising as fast as we are doing research. So questions are also coming fast in a faster way. So that will broaden our idea to develop that cultivar that is suitable, that is functional, that can we grow in fields, that is having public acceptance, all those things that are coming. So we have to prepare ourselves like that. So it's good to have that approach in advance.
Right. According to you, which sectors of plant science research will prosper in the near future?
It's a really tough question because this is not the thing that which branch will prosper, because all the branches are prospering very well and people are doing very good. Some scientists are doing very good in their respective fields. Even if you say in physiology, some scientists are there who are doing very good physiology. They are having so good coming some with some new prospects in physiology also, and in biochemistry also, in chemistry also, in physics also. We are several new arenas are opening day by day. So we do not know the same thing as in previous question, what is hidden in the future. So scientist has to do always has to search something for betterment of the society and some novel things that came to their minds, and while doing research some more new things came to their minds. So the thing is that now the current scenario is that the climate is changing and the population is increasing, so we are focusing on that respect. And now certain these bio attacks are there, so now people will think from in different manner. So from now onwards, from last year onwards, like the people living standards are changing, their purchasing habits are changing, their eating habits are changing, all these things are now people are focusing more on immunity things. Now the scientists will come up with more immunity boosters crops. Now people are taking medicines, why not we have to we have them in the crops plant. Then the scenario will change as the problem will come. So which branch will better deal with that,
Right.
but we have to do research.
Definitely. You have received many prestigious fellowships like the SARB Young Scientist and Dr. D.S. Kothari Postdoctoral Fellow, to name a few. Could you please share with us what was your strategy?
Yeah, behind all those these fellowships, I will like to thank my mentors who really used to go for these fellowships, and like Dr. Singla Parik, that boost me. She boosts me to apply for this SERB Young Scientist Award, and also she boosts me further for applying for DS Kothari postdoctoral fellowship, to have my own fellowship and to start my own research ideas and implement them. So with their ideas and some of my own, and by their boosting, I came up with these all these fellowships. So I really thank to them. They are really good mentors for me as far as.
Okay, you are the reviewer for many international journals. Kindly give our viewers some tips to improve manuscripts.
Yeah, these days so many manuscripts are coming with different approaches, and people are doing really good research. Some people are doing really good research. they are very hardworking, and some groups are really hardworking. But apart from that, there are several researchers who just don't go through their research very well before sending it to publications. Like a few points you have to remember that your while acceptance of publication, the writing part is the basic thing. Is it the selling thing what you have done in your research? You know? So you have to explain some unknown person about your research, what you have done, and what is the usefulness of that research. So writing part is you need to be very well written, because several reviewers they found the grammatical errors and all those types of things in while reviewing the articles. And also the what you you are saying in your research, like in from in your title, that comes and in your research also. Sometimes people make so broad titles, and they have done so only doing one study and then saying, oh, this will change the whole scenario. This will means distract the reviewer to also to accept the your articles. And the another thing is that whatever you are saying, that is or what you are means writing in your article, that is to be supported by research also. So you are
Mm.
fault
Right.
tolerant, so you have figures for that, you have to analyze for that different graphs and all those things, and real field pictures people will see that that are always convincing. So
Mm.
always so those people who will give original research and some identify some mechanism also, then the reviewer are always interested in having accepting those research,
Mm.
rather
Right.
than those people who are just writing and making some tables or figures and not giving actual experimental figures what they have actually done.
Right.
These days I am seeing that most of the around 80% of the manuscript they are hiding original figures, just making these graphs and like that tables and then sending. Oh, so, you have to keep in mind what you are actually saying, and you have to give the data for your support. As much data as you will give in your support, that will convince the reviewer finally to accept that your research, because
Mm.
every journal wants the good article to come to their part, because finally it will affect their impact factor also. So
Mhm.
the so this is the main thing, that if you will do good research, so reviewers are already been interested in your research. They will try to incorporate your research in their journal, because then only the journal impact factor will increase or increase, that finally every year now the impact factor will come. So this is
Oh, that was the great tips. Thank you so much. Finally, uh, coming to the end of this interview session, what would your words of advice be to the students and scholars who are new to the field of plant science research?
and the only advice is that be consistent and don't lose hope. Anytime you need not to lose hope that you are doing good research. Sometimes the people, our students also, that they lose hope that what the research we are going, are we going in the right direction or not.
Mm.
For that, you always try to read more and more about your research related papers, and get an understanding, clear your fundamentals more and more. The more you have good fundamentals and more you believe in your study. First of all, the major thing is that you have to believe in your own study also. This is
Right.
very important, that while doing research you have to believe that yeah, this is the research you are doing is and the another important thing is that you have to be focused toward the society, that yes, I have to do something for society, for the poor farmers that are sitting in their fields thinking that one day we will release some variety or that thing that they can use in their farm to increase their income, because
Right.
they are really poor. Some are really poor, and they are some are really hungry. Some are not having a single diet a day. So they are waiting for something, some person to do something for them. So this is our responsibility basically. So you have to understand your responsibility and never lose hope. One day you can do something, but by the time you have to make your understanding, good understanding, that if you think that you are lacking in some area, then try to fill up that area. That will help you in later research also.
Hmm. That was great advice. Thank you so much. It was wonderful to hear about your views. So let's go back to Shubho now. Shubho, over to you.
Thanks, sir. It's a nice conversation, and am not include few questions. Those are came from the students, but I like to discuss one or two with you. Like, is that India is a good place for PhD?
Yes, why not? People have done very good research in India also, and these days some students are doing very good. Even if you will see that the people that those are in top positions in India, if you will talk to them, you will definitely found that they have not only they are in top position, but they have really done good in their research and academic career also. They worked really hard to reach and that those positions. So it only depends upon you, because people can do good research anywhere, and the only thing is the idea. The basic thing is idea, that is IPR. Your IPR basically. So good to your ideas, you can do good research. That's not the thing that you have to do the costly science, that the you have to spend so much on the chemicals, and then you doing the CRISPR-Cas directly and all those things to do a good research. Rather, you can do simple experiments like physiological, biochemical experiments also to do good research. On all the papers that are coming in science, when you will see that you will found that besides having developing, like we are developing transgenics or do CRISPR-Cas, we have to later prove it through physiological and biochemical approaches. So this is the thing that in the high impact journal also, if you will see, there are transgenic papers also, and then some physiology and biochemistry papers also. Very simple theories they have done, but they have really a good ideas. They have not the big costly studies. So anywhere you can do good studies, good researches.
Okay, sir. And others questions are, I'm sure these answers are available on online, like protocol related questions or the good institute or fellowship. Some people are asking from Africa, there is any fellowship for India to doing their research. And
There is a TWAS fellowship that they can apply.
yes.
Yeah, good to do research in India. TWAS.
Mhm. And sometimes, like ICRI said, they are also doing and giving the fellowship in some personal proposal. I'm sure. I'm not.
ICGB also has a fellowship.
Yes.
ICGB also has a fellowship. Very good fellowship. Artro fellowship, I think. That is for African countries and poor countries to come to do good research in India.
And many people from Pakistan, they are very interested on transgenic research, but their concern is that when it will be marketed in Pakistan. So it's very regional, it's country specific manner. Some countries are already doing and cultivating the transgenics. Some countries are going to be cultivated, like Bangladesh and Pakistan, related to golden rice. And for India, we're cultivating only the BT cotton. But in future, there are a lot of pipeline, and nowadays the policymakers are both very active to doing that, and CRISPR give a hope to doing research on transgenic in a different manner. Many people are interested to know about the transgenic. Those are, I request you, there are few articles recently published in Plant Biotech Journal and others. Those are actually data driven and original scenario about transgenic research. Go for that. It will give you some boost, because most people are taking it as negatively initially because of lack of knowledge or information, but nowadays information is vast and many papers have come to that. One asks that whether is any problem to environment to cultivate the Tanzania. Actually it's under study still, but still those are published. They are not showing any environmental effect on Tanzanic plant growing on the field. But for long, it's already 20 years in case of golden rice is published and is cultivated many ways and many seasonal. So there is nothing visible significantly level. But it's obviously when we go for the Tanzanics, we expect more other than all the aspects. Either we focus on the yield, either we focus on the biomass. So sometimes we neglect few of the parameters. Those are the insignificant for the research. So obviously, Bioingene is a platform where you can discuss, but this discussion cannot never end in a one day or one session. So keep touching. We are try to build such families where we can ask the question to the persons and to the platform. Thanks, Joshi sir, for today's webinar.
Dr. Subrato and Dr. Soma and Dr. Sharma. And I also thank to all the viewers and for asking questions and listening my talk. So thank you very much. Please give me my your comments on my mail ID that I have shared in my slides, or you can call me directly on my phone. So thank you, sir.
Thank you.
Now I request Soma to give some little bit tour about a Bioingene PhD journal and how they can collect the certificate. Sir, you may end your sessions. We need 10 minutes more. Okay. Thanks, sir. Thanks all the participants, and those are waiting for the feedback link. You need to send me a request on our email address, and you need to copy the password from our chat box. We are not giving the password in any other methods. And those are waiting for next registrations. Just go our website is bioingene.com. There always you can find some upcoming webinars in that website. You can register yourself and you inform your friends also. And I request, if possible, please subscribe our channel. It will make you updated. If you miss some webinar, it prompts you that when we are going to live. I request Soma to give a small tour on our website and where you can find your certificate after 3 working days. I request Soma share your screen and go for that.
So this is the website, bioingene.com. As you scroll down, you will see that we have our upcoming webinars lined up over here. Our upcoming webinars, you can find our upcoming webinars, the information over here. You can register by clicking the register link from here. The Google Forms will open and you can register from our website for the webinars. Okay, so when we scroll down, we can see that here are forms for to be a speaker on Bioingene. You can apply for that, and also at the bottom of the page you will find that here is our latest PSJ issue. That is a Plant Science Journal issue from Bioingene. the mini reviews, articles, perspectives. They are all, you can find them here. The PDFs are here. You can view them. Also, if you go to the top of the page of our website homepage, you will find that there is a tab for in magazine. There's a magazine tab. If you click on that, so you go to our Bioingene Plant Science Journal page. This is our Bioingene PhD page. You can get all the information you need if you are thinking about submitting a write-up, which could be a review, a mini review, an original research, a summary of an original research, or your perspective about something, about a current any current topic, or anything that you think that can be a thing in the future. Any perspective, that is your original writing, definitely. And you will find all these informations on this website, on this page. Then if you further scroll down, you will find that there's a link for submitting your manuscript. So you can submit your manuscript here. All these, like if you're review, mini review, or summary, whatever, you can submit over here. And then you scroll further down, you can see that the already published articles are on this page. Okay, so I think, and if you have any more questions, you can always email us. If you're thinking about writing and you find that you need more guidelines, you can always contact us. We are following a very strict peer review protocol for our journal. So your original research or review, whatever you're submitting, that will be peer reviewed. And I suggest you always use academic English and proper English for writing your articles. And I would like to ask you to please very carefully, when you're writing, please be more careful about your references, because when you're writing, like sir said, like Dr. Joshi said, he gave some wonderful tips in the interview section about writing manuscripts. So there was one tip where he said that when you're writing something, when you're putting in data, when you're putting in information in your article, then you should always back it up with a reference, because without a reference your article means nothing. Whatever you have written, what that sentence, those sentences will mean nothing if they don't have a reference to back them up. So you will need to put in the reference, and then you need to definitely put that detailed reference in the bottom of your article, in the heading references. You don't forget about that, because we have been receiving some articles where you forgot to put that in the reference list. So please keep that in mind. Please check your whole manuscript, check your whole article before submitting, that you have put in the references in parentheses in the text, and then the details of that reference at the bottom in the reference section. Another thing which some of you are facing, the younger writers are facing a little problem with the images. So you are putting in some images that you have not mentioned the sources. See, when you're putting anything in an article, those things must be backed up with the sources. When you're writing something, if you have not given a source, then that means that that is original, that you have found that out, or you have taken the picture. But if that is not the case, it's a third party picture, third party image, third party table, whatever it is. If it is third party, if you've taken it from somewhere, then you have to, it's a must that you have to mention the reference, the source of that image, and the detailed reference in the reference list. That, all right, so to sum it up, you can submit your review, your mini review, original research, Like if you are doing your master's dissertation or thesis, you have done some original research in your college or university lab, you can definitely publish that on Bioingene. So write it up in a good format, and we follow Frontier's reference format. You can get that online. And we also, anybody can write a summary. We are accepting summaries are very interesting in the sense that you read a paper and then you grasp it, and then you put in your thoughts and you explain it in a very simple and easy language. So when writing summaries, please keep in mind that it needs to be very simple English, so that everybody can understand. Always keep in mind that when you're writing a summary, that it is a link between the non-scientific and the scientific world. So non-scientific people can also understand when they're reading a summary. So that one. And then perspectives. Your perspectives are that when your personal views on any topic. Okay. So all these we are accepting all these articles, and then comes the certificate collection for the Bioingene webinar. I'm going to say a little bit about that, the certificate collection. So what you can do is that go to our website. You can see the website here. You can go to the website homepage, and at the very top of the page, there is this green tab, collect your certificates. You can click on this and you can collect your certificate from here. So you can see that the name of the webinar topic, the webinar topic name, and the date is here. All are given over here. So you can go and you can search which certificate do you want, and you can just collect the certificate from here. So today's webinar will be, you can get it from here, and you can just click get certificate. So you can collect it from the shared Google Drive. Is there anything that needs to be said, or I'll just go back to you now?
Thank you. Those are asking for the fees. We don't charge any fees. It's totally open for all. No submission. Those are authors don't need to be pay any fee, and those are readers. It's absolutely free. Actually, our all activities are free for all and open to all. So there is no charge. But obviously, it's a peer-reviewed channel. So you send something and we're going to publish. It's not happening here. We are going for the peer-reviewed process. It took generally 60 days to complete submission to publish your journal. So we follow a strict rules about peer-reviewed, and we help authors in different ways, like improve their English writing, improve their content, improve the structure of journal. These are our responsibility we took in our publication team, through our publication teams, and our teams are well qualified and have the PhD degree on that. And for our reviewers, those are interested can email us. Then we give you a form for enrollment. You need to be a degree in PhD degree and experience in international publications. You have 4 or 5 papers on your hand. It's our eligibility criteria for being a reviewer for Bioingene PhD. So thanks for everyone for attending today's webinar. Registration for upcoming 2 webinars are ongoing through our website. Please follow us in different social media. We're in LinkedIn, we're in YouTube, we're in Facebook and Twitter. Thanks everyone. If you need, still waiting for the feedback link, you need to email us, request for the feedback link with today's password. We send you the link immediately. Thanks.